Welcome and thank you for standing by. I would like to inform all participants that your lines have been placed on a listen-only mode until the question and answer session of today's call. Today's call is being recorded. If anyone has any objections, you may disconnect at this time. I would like to turn the call over to Stephanie Sherholtz. Thank you. You may begin.
Thank you. Good afternoon. I am Stephanie Sherholtz from NASA's Office of Communications. Thank you for joining us today to talk with our experts about science investigations that will launch alongside the astronauts in Crew Dragon this weekend. As a reminder, NASA's SpaceX Crew-3 is targeted for liftoff in the early hours of Halloween on Sunday, October 31st at 2:21 a.m. Eastern Time from Launch Complex 39A at NASA's Kennedy Space Center in Florida. The Crew-3 flight will carry NASA astronauts Rasha Chari, Tom Marshburn, and Kayla Barron, as well as ESA astronaut Matthias Maurer. The crew members will join Expedition 66 aboard the International Space Station for a 6-month science mission. The Crew Dragon with the astronauts aboard is scheduled to dock to the space station about 22 hours after launching. So docking will be about 12:10 a.m. Monday on November 1st. So here to talk with us today about some of the research launching with the Crew-3 astronauts and the investigations they'll support are David Brady, who is our Associate Program Scientist for the International Space Station Program at Johnson Space Center, who will discuss how the Commercial Crew Program is boosting research aboard the orbiting laboratory. Dr. Jason Stagno from the National Cancer Institute's Center for Cancer Research, who will discuss the Uniform Protein Crystal Growth Experiment. This experiment will launch with the Crew-3 astronauts, and it will return with the Crew-2 astronauts when they return to Earth no earlier than November 4th. Dr. Grace Douglas, the lead scientist for NASA's Advanced Food Technology Research effort, will discuss the food physiology experiment. Dr. Hector Gutierrez, Professor of Mechanical and Aerospace Engineering at the Florida Institute of Technology, will discuss the smartphone video guidance sensor investigation. And Dr. Gilles Clement from NASA's Human Research Program will discuss the standard measures investigation. Our speakers will provide a brief overview of their research, and then we will open the lines for a few questions after each speaker. If we have time at the end, we can take additional questions. Your phones are on mute, and to get into the question queue, you can press *1 on your phone at any time. We will call on you, and the operator will open your mic so you can ask your question. We do ask that you please identify yourself and your media outlet and stick to one question. If we have time, we'll allow reporters to ask a second question. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 888-566-0076. Again, that number is 888-566-0076. The recording will also be available on the NASA YouTube on which this is playing. So to start, we will hear from David Brady, the Associate Program Scientist for the International Space Station Program at NASA's Johnson Space Center in Houston. Over to you, David.
Thank you, Stephanie. Well, welcome, everyone. Thank you so much for being here. We've all gathered to see Tom, Rasha, Kayla, and Matthias begin the most visible portion of their journey. And as part of that, much of what you'll observe over the next few days is associated with what our team likes to refer to as answering the what and the how questions. Well, at this event, we're going to focus on providing a few answers to the why question. One of the benefits to ISS research that's provided by our still relatively new Commercial Crew Program capability is being able to launch science alongside the crew. And by being able to do so, we've increased our ability to conform to schedules that are based upon the needs of our researchers, which makes for better science. For example, as Stephanie briefly mentioned, the Uniform Protein Crystal Growth, or UPCG investigation, which you will hear more about shortly, will be going up with the Crew-3 crew. and then returning next week with Crew-2. Another advantage of our CCP capability is that it keeps the transportation path close to the lab. For the Dragon vehicle, science is launched from Florida and recovered just off the coast of Florida. And using UPCG samples as an example again, they are samples that need to be expedited back to the investigators, in this case, for X-ray free electron laser or XFEL imaging. Now, on a slightly different tack, I also want to mention how Crew-3's support of ISS research and technology is instrumental for testing new, upgraded environmental control and life support systems, or ECLIS as we call it, technology that is crucial for our future exploration missions. This includes items such as the Universal Waste Management System, or UWMS, or the toilet, the new toilet as you all have come to know it, the brine processor assembly for water reclamation, carbon dioxide scrubbers like thermal amine, and the 4-bed CO2 scrubbers, as well as 2 new hydrogen sensors that are slated to arrive aboard the SpaceX Cargo Dragon CRS-24 mission in late December. Now, these are just a few examples of space station science and technology that will be conducted by the Crew-3 crew. And I'd love to go on and on about more examples to include some of the other research examples you're going to see today. But as I've said in past science telecons, I'm just the warm-up act for the main event. So let's get to the rock stars of ISS research. Thank you again for your interest.
Thank you, David. Now we will hear from Dr. Jason Stagno, who will discuss the uniform protein crystal growth investigation that David Brady referenced. Dr. Stagno is an associate scientist in the National Cancer Institute's Center for Cancer Research, Protein Nucleic Acid Interaction Section. He is a structural biologist with expertise in X-ray crystallography and molecular mechanisms of gene regulation. Go ahead, Dr. Stagno.
Thank you. So, hi, everyone. I'll state right off the bat that our science is a little nuanced and complex, so I'll do my best to give enough detail to understand what we're doing, but not bore you with too much detail. So the goal of our experiment ultimately is to grow tiny crystals of RNA, ribonucleic acid, under microgravity, and then diffract those crystals here on Earth using an extremely powerful light source called an X-ray free electron laser, or XFEL. This method overall is called X-ray crystallography, which enables us to determine the three-dimensional atomic structures of important molecules like proteins, DNA, and RNA. And most of you might be familiar with scientists Watson and Crick, who took the first X-ray photograph of DNA in 1952, although it was Rosalind Franklin who determined the 3D structure of DNA shortly thereafter using as well as in engineering of RNA-based therapeutic switches. And riboswitches are called switches because they turn genes on and off through a series of structural changes, which are controlled by the binding of a small molecule. Perhaps this is an oversimplified illustration, but think of turning a light switch off and on The RNA is like the light switch, which can be either up or down, the position of which determines whether the light is on or off. And the switching mechanism is controlled by interacting with something else, in this case, my hand. And the physical movement of the switch up or down is analogous to the physical changes in the structure of the RNA that turns the gene on or off. So like a camera with a very fast shutter speed, we're trying to catch snapshots of the RNA structure as it changes in response to stimulation, to record a molecular movie, as it were. And for this purpose, we need billions of very tiny crystals rather than a few large crystals, which is often the focus of other crystallization experiments on the ISS that have been done in the past. Crystals with such small volumes are more likely to accommodate the molecular changes in the lattice with a higher degree of synchrony. However, we need crystals to be as homogeneous and of the highest quality possible in order to get the optimal diffraction data before, during, and after the switching mechanism. And the hope is that microgravity environment for crystal growth might accomplish this. As was stated previously, the experiment will be done almost immediately after Crew-3 arrival, and will return with Crew-2 a few days later, and then the diffraction experiment will be done immediately after that. So I'll be happy to take any questions at this time.
Okay, great. Thank you. That was a very good description. Again, if reporters have a question, please dial *1 to get into the queue, and we'll start our first question with Russell Pounds.
Thanks, Stephanie. Dr. Sagno, thank you for that description. And, you know, we all know that this kind of science is about getting the good funding and getting the public involved. And, you know, I'm not a scientist, but I got the sense of what you were trying to do with that. So I'm curious about, when delivering the presentations, when is the aha moment for understanding the value of investigating this kind of science for the business community or the public when you go out to talk to them? Thanks.
Sure. Yeah, I mean, the aha moments experienced in this type of research are when we can see the diffraction quality of the crystals grown under microgravity are better, and the data are more interpretable and used to generate information that otherwise could not be obtained. So this is a very nascent field and has never been done before in this context. So we, of course, you know, we can hope for the best, but if this does work, we can really pave the way for further time-resolved structural studies that will allow us to kind of see molecules in action, being able to elucidate their molecular mechanisms. And this has all kinds of implications for biology and for molecular therapies.
That's great. Thank you so much.
Okay, moving on to Ken Kramer.
Hey, thank you for doing this very interesting experiment. I'd like to know why, however, you have such a quick turnaround here. A lot of times you need longer time to develop crystals, and you're going to only have about a week or so. So I'm curious about why that is. Thanks.
Sure. Yeah, that's a great question. That has a lot to do with the science and the method that we're using. So conventional crystallography is usually done using vapor diffusion techniques, which require the allowance
Thank you.
Mm-hmm.
Thank you. And can you tell us how the growth is initiated?
Yeah, so it's actually very simple, which is great for the astronaut to perform the science, in that we have two liquids, one of which is the RNA, and then the other liquid is a precipitating agent, which induces crystallization. And those are in individual syringes that are the path of which between them is blocked. So the astronaut will remove the syringe pair from cold stowage and allow it to thaw to ambient temperature. And then the experiment is initiated simply by the pushing or mixing of the fluids through the syringe pair, and then incubating the sample in a 22 degrees C incubator.
Okay, that's fascinating. Thank you.
Mm-hmm.
Great. At this time, we'll go ahead and move on to our next speaker, who is joining us. So our launch is targeted for Halloween, and while that's a day known for indulging in sugary treats, we can't forget the importance of a healthy diet. So talking to us next is Dr. Grace Douglas, who's our lead scientist for NASA's Advanced Food Technology Research effort, and she will discuss the food physiology experiment. Her work focuses on determining methods, technologies, and requirements for developing a safe, nutritious, and palatable food system that will promote astronaut health and performance during long-duration space missions. Over to you, Dr. Douglas. And again, as you're listening to her talk, if you have a question, please press *1 to get into the queue. Go ahead.
All right, thank you. I'm very excited to get this opportunity to speak to you all today about our study, which is looking at the impact of diet on the human immune response, the gut microbiota, and nutritional status during adaptation to spaceflight. And there are multiple co-investigators on this study with expertise in each of these areas. So it's a very integrated study. And the reason that we wanted to start the study was because when we look at crew in spaceflight, we learn a lot. And some of the things that we learn are that they do have changes in their immune system. So some dysregulation of the immune system, reduced immune cell function. They do continue to lose body mass even over these 6-month ISS missions. We do still see body mass loss in some crew. And we also see things like increases in stress response. So as we go further into exploration and these missions get longer, we want to be able to provide countermeasures that are going to be on these vehicles anyway, because our potential to provide medical countermeasures might even become more limited. And diet is a great one of these. So if we look at diet, it impacts every aspect of our physiology. What we eat, our microbiome eats, it produces other metabolites, it's interacting with the immune system in our gut, it influences the gut-brain axis, so it's basically influencing everything. And if we're eating the right diet, we can actually promote health and performance. And so we want to start understanding, specifically with the spaceflight diet, which by nature has to be more processed and shelf-stable, how is that impacting the microbiome, the immune system, the nutritional status over time, and can we improve upon that? And we can start to understand that by looking at the resulting markers from different biological samples. And so our goal here is to increase the amount of certain foods that crew have access to, and also to give them that choice to find the foods that they want to eat from the healthier foods that can provide a greater selection of fruits, vegetables, and foods rich in omega-3 fatty acids, lycopenes, and flavonoids to those crew members, and then determine its impact on immune system function, the microbiome, and nutritional status over these International Space Station missions. And ultimately, this can start to provide evidence-based recommendations for dietary countermeasures in spaceflight to enhance crew health and safety on exploration missions. This becomes very important because some of the healthier foods are actually the least dense foods, and yet when we're looking at these missions, resources is a big concern, and food takes up a lot of resources, and so we have to be able to provide the evidence that says these better food systems are providing these benefits, and then we can make better trades between resources and how we're planning for human health and performance. So I think that's probably where I'll stop and ask if anybody has questions.
Great. Thank you, Dr. Douglas. And we are aware that the call has dropped a couple times, and we've gotten it back for folks who are listening on nasa.gov/live. We will have a recording of the call, as was mentioned previously. Again, about 1 hour after the call concludes, you can listen to a replay by dialing 888-566-0076. And if you are listening online and have a question you want to submit, you can send it to @ISS_research. If you miss a bit and want us to repeat or revisit something, please do let us know on Twitter or social media. And with that, we'll go to the question queue for Dr. Douglas. Paul Brinkman, go ahead.
Hi, yeah, this is Paul Brinkman with UPI. I was just wondering if you could kind of summarize what you've learned already through ongoing research along these lines at ISS. And then you mentioned a few selection of fruits, vegetables, and richer omega-3 fatty acid foods. But I'm wondering if you can give us any specifics about exactly what kind of food is going up to ISS in this mission. Thanks.
Sure. So the first part of that was what have we learned so far? And so we have just implemented this recently on the International Space Station. We are expecting to see benefits. We did previously put these diets into the Human Exploration Research Analog missions on Earth for 45 days, and we did see improvements even in 45 days in several nutritional outcomes and even in some cognitive reaction outcomes. And so over 45 days, I can say one of the very apparent outcomes was a reduction in cholesterol and some other nutritional benefits similar to that. So pretty quickly you can start seeing benefits just from increasing these numbers of fruits and vegetables and things like that. And the very interesting part of this is that we are doing such an integrated study. So we're collecting our biological measures at the same time. We're looking at all of these outcomes at the same time. And we are collecting very detailed dietary intake because these missions do not have the potential to introduce outside foods. We actually have a better capability to collect very detailed dietary intake compared to a lot of studies on Earth where it is a lot easier to access outside foods. So this is a very integrated study that's unique, and for the length of time that we're doing it and collecting these detailed dietary intakes, we expect to start seeing some outcomes in spaceflight that are not only related to nutritional and cognitive outcomes, but also related to improvements in immune outcomes. So that is our expectation. And we have also seen greater stability in the microbiome, which can actually indicate a more robust microbiome that will be more robust against perturbations from illness. So the second part of your question was what kinds of foods are we flying? So we do fly a processed food system. By nature, we have to provide foods that are shelf stable. However, within a processed food system, you can still provide healthy fruits and vegetables and fish type products. So some of the things that were developed specifically to support this study were some more freeze-dried vegetables. So there's like a butternut squash and sweet and savory kale, and then also some extra fish products were developed. So a lot of the options that are available that are processed are things like salmon and tuna. So we developed some barramundi options, which is a fish that's farmed to be high in omega-3 fatty acids, and it tends to have less of a fishy flavor, so it can be an alternative to some of the options that are available in processed form that meet the requirements for spaceflight food systems.
Did we lose Stephanie? This is David.
Yes, thank you. Can you If anybody else has questions, please do dial *1 to get into the queue. My question for Dr. Douglas is, what input do the astronauts offer on meal selection for their missions?
So for this evaluation, one of the things we're really trying to understand is, can we improve intake? So we do, as I mentioned, we have a good variety of food on station, but we want crew to be able to get the nutrition they need from foods that they want to eat. That becomes very important. Food becomes very important psychologically as well with long duration. So we do meet with crew ahead of these, their missions, and we try to provide the healthier food options that they personally want for their participation in this study. So when they're choosing, they're going to eat a higher quantity of the fruits and vegetables and fish during their segments of the mission. They do get some choice in what is included in that, and we work with them so that it meets the study requirements. And then we work with them throughout the mission to try and make sure that they're meeting those targets for the study.
Great. Thank you. At this time, we don't have other questions in the queue for you, so we will move on to our next speaker, who is Dr. Gutierrez, Professor of Mechanical and Aerospace Engineering at Florida Institute of Technology, who will discuss the smartphone video guidance system. Please go ahead, Dr. Gutierrez.
Okay, thank you. Well, first of all, thanks to everyone for the opportunity to present our research here on this public forum. Very excited and happy to be part of the upcoming flight with Crew-3. We have been working with NASA Marshall on the development and demonstration of SVGS for many years now, since 2017. They started even earlier. This is a technology that was originated at NASA Marshall, and has been already deployed on different versions on other space missions. But the initial push on SVGS, and that remains on its name, was to demonstrate that it's possible to have a software sensor that can be deployed on multiple platforms to calculate the position and attitude of illuminated beacon. And that is a very valuable means to provide guidance and navigation information when you are doing proximity maneuvers with a small spacecraft or drones or mobile robots. So although SVGS has a smartphone on its name, that has been one of its implementations. So in one version of SVGS can be deployed using a cell phone, and therefore the code can be programmed to run on a cell phone as an app that uses the phone camera to take photos of a target and navigate and obtain position and attitude information from two-dimensional images coming from the phone. So starting from the beginning, SVGS is a technology to calculate position and attitude of a spacecraft. It requires a CPU and a camera to take photos of an illuminated target. And that provides this technology huge flexibility to be deployed on any kind of hosts. Most robots will have multiple CPUs, and many of them will have cameras. So whether you use a smartphone or not, SVGS is a technology that can be deployed on a variety of robotics platforms. And we're going to demonstrate this by deploying it on the free-flying robot existing on ISS, which is called Astrobi. So maybe some of you are familiar with it. Astrobi has been already active on ISS for more than a year, and is replacing the previous free-flying robot called Spheres. Astrobi is a very advanced robotic platform that is used to support many different kinds of science experiments. And in this case, we're going to use it to demonstrate the deployment of the SVGS sensor as a software sensor running on Astrovis hardware. And what we're flying in this Crew-3 vehicle are going to be 4 LED beacons that will be used to implement a number of maneuvers on ISS as part of this demonstration of SBGS. So in summary, SBGS, although it has a smartphone on its name, the ISS mission does not include using any smartphones. It's just running the SBGS software on the host, in this case Astrovis, to take measurements of position and attitude and to use those readings to provide waypoint commands to the robot. So the main objectives of the mission are, number one, to demonstrate that SBGS can be deployed using resources of a robot host, in this case Astrovis. Number two, we want to show that we can measure position and attitude with precision and accuracy that are comparable to the metrology that is already existing on Astrovis. Number three, we want to demonstrate that it's possible to identify multiple spacecraft in a formation flight maneuver by using different color of LED beacons. And finally, we're going to demonstrate the use of SBGS to orchestrate and provide motion commands in formation flight maneuvers for multiple Astrovis. So in a nutshell, that is the purpose of our mission, and we're going to be having operations between February and July of next year, and typical experiments should last less than an hour, somewhere between 15 to 20 minutes to an hour. The maneuvers are in increasing complexity from 1 to 5. And so we're very proud and happy to be flying to ISS on the Crew-3 mission in a couple of days. And from there, I'll be happy to take any questions if there are any.
Great, thank you so much. So it does sound like for some of the investigations, at least all 3 SOBs will be flying at the same time. Is that correct?
Yes. On the more complex maneuver, maneuver 5, there will be 3 Astrovis flying in a formation flight maneuver. The other maneuvers include only 1 or 2, at the most is 3, yes.
Okay, great. We'll go to our first question in the queue from Lauren.
Hi, I was trying to get this in for Dr. Douglas, actually, so I'll ask it very quickly if she's still on. I just wanted to confirm that that all crew members are going to be participating in sending over information for the diet. I'm sorry, can you please repeat that question? Yeah, I just wanted to confirm that all members of Crew-3 will be participating in the space flight diet and sending in information back. So at this point, we don't disclose who's in the studies unless they disclose it. Thank you. You're welcome.
Thanks, Lauren. Our next question is from Mark Carreau.
Thank you. This is Mark Carreau from Aviation Week for Dr. Gutierrez. I think when I read the background, I think I read this correctly. There may be some application to the human landing system for some of the work you're doing with the SVGS?
That is absolutely correct. One of the potential applications short-term is to demonstrate SVGS as a support technology for automated landing on human landing operations. In fact, right now we have an ongoing project supported by NASA Marshall at Florida Tech that is demonstrating the use of SVGS to do an automatic landing from 120 meters altitude, in which SVGS is used for first approach from 100 meters to 50, and then from 50 to 20. And then another fundamental difference when you do planetary landing is that you don't have GPS. And a huge amount of the research that has been done so far with drones, for instance, in landing, are based on the availability of GPS. So SBGS is envisioned as a technology that can be used in planetary landing for an approach phase, let's say from a few hundred meters altitude down to maybe 20 meters. And then after that, some kind of inertial navigation is needed, since the dust will make any kind of image processing very, very challenging on a typical planetary landing situation. But yes, it is definitely being envisioned as a technology for planetary landing, and we are already working on the details on how to demonstrate SBGS ability to work on such environment.
Okay, thank you. Our next question comes from Paul Brinkman.
Hi, yeah, it's Paul with UPI again. For Dr. Gutierrez, can you just sort of describe how the sensors and the beacons are deployed? My understanding, which, maybe I missed something, but I guess the sensors are on the robots themselves, and then the beacons are deployed throughout ISS, or in a confined area, or what portion of ISS are we talking about? Thanks.
Okay, very good. Thanks for your question. So the SBGS experiments are going to take place at the JPM, on the Japan Pressurized Module. And we have two basic configurations in which the beacons are attached to the rails on JPM. And in two of the experiments, we're only going to have one beacon attached to the wall and one beacon attached to another astrobeam. So, for instance, on maneuver number 1, you have a single astrobeam navigating relative to a beacon attached to the wall. On maneuver number 2, you have one beacon. We have one beacon attached to the wall and one mounted on the payload bay of an astrobeam. So we'll have a leader-follower maneuver where the leader is going to be moving relative to the wall beacon, and the follower is going to be looking at the beacon installed at, the undercarriage of the leader. So in more complex maneuvers, we'll have up to 4 beacons on the wall of the JPM module, and in maneuver number 4, for instance, there will be 2 on the wall and 2 on 2 astrobeams. So there are a number of configurations. I wish this would allow that screen sharing. It's a lot more easy to explain this with pictures, but in a nutshell, some of the maneuvers involve having the beacons attached to the wall of the JPM. Some of them involve having them attached to the payload bay area of astrobeam, and some of them are combinations of both.
Okay, great. Thanks.
Thank you.
Thank you, Dr. Gutierrez. And did you say how many total maneuvers you are planning to embark on?
Yes, there is a total of 5. It may turn into 6, but right now we're looking at 5 maneuvers.
Okay, great. Thank you. Okay, not seeing any more questions in the queue at this time for Dr. Gutierrez. We will move on to our next speaker. So we will hear from Dr. Gilles Clement. Dr. Clement received doctoral degrees in neurobiology from the University of Lyon in 1981 and in natural science from the University of Paris in 1986. He has 40 years of space research experiments, flown on Soyuz, Mir, Space Shuttle, and the International Space Station, and he currently serves as project scientist on the Standard Measures Cross-Cutting Project for the Human Research Program at NASA's Johnson Space Center. Dr. Clement?
Thank you, and thanks to everyone for giving me the opportunity to present our research project, which is called Spaceflight Standard Measures. So what it is a subset of measures that the NASA Human Research Program has chosen to systematically collect astronauts until the end of the space station. And so why we are doing this is the objective is to gather as much evidence as possible for mitigating human health risk during exploration mission. The NASA Human Research Program has identified a series of health risks for humans during those exploration missions that could be caused by altered gravity, radiation, isolation and confinement, and as well as different spacecraft design. And our project, Spaceflight Standard Measures, address about 1/3 of this risk, which have been identified. What is different with this project is that it's a cross-cutting project. And by that, we mean that we collect response not from just one discipline like muscle or bone or immune system, but we collect response from all of the disciplines at once, in physiology, human biology, and psychology as well. So the STANS team for this project is composed of the lead scientists in these research disciplines at NASA Johnson Space Center. So what are those measures that we collect? We have a portfolio that includes, as I said, measures from various disciplines. For example, we measure the duration of sleep of the astronauts using very simple smart watches that they wear 24/7 over a period of 2 weeks every other month during the flight. We also ask them to complete monthly surveys about how they interact with their team to evaluate team cohesion and performance. We also have a laptop on the space station in which we can test them over 10 different neurocognitive tests to measure their reaction time, their attention, concentration, working memory, spatial orientation. In the 20-minute test, we can evaluate all of these cognitive performance. We're also collecting some blood and saliva samples to look at the immune status and function and general blood and urine chemistry at different periods during the flight. And the astronauts also collect some body swabs, and this is to measure the concentration of various microbes on their body when they are on board the space station for long duration. And finally, we also have tests that we do only pre and post-flight to measure, for example, the cardiovascular structure and function. We do an ultrasound examination of their carotid artery, looking at the thickness of this artery, which is a measure used on the ground to predict cardiovascular events, and which is also an early marker of artery damage that could be induced by radiation. And finally, we also look at the sensory motor function, how they are able to stand up, to walk immediately after they land. And we have tested in the field, in that case, at the Riley Airport only 4 hours after splashdown when they return. And we will test how they can stand up, walk through an obstacle course. What happens if they fall? Can they stand up? What is their cardiovascular reaction to this change in posture? And this is to evaluate what type of critical task the astronauts can do after having been in space for 6 months. So this project, the data is collected before the flight, as I said, and after the flight. And during the flight, they are collected 2 or 3 times during the flight. So those are really a limited subset of measures, but they are standard. That means we repeat always the same measure, and that's to compare across mission with various duration, with various countermeasures. The project has began in 2018, and we have already collected measures on 15 subjects during missions lasting from 2 weeks up to almost a year. And the same measures, with the exact same methods, are also collected during analogs of space flight, such as in bed rest, long duration bed rest, and in isolation and confinement studies, also for various duration. And once we collect this data, then we place them in the NASA Life Science Data Archive, and where they are available for other investigators. For example, during this mission, we are sharing data with Dr. Douglas as well. So they are available for the other investigators to interpret their own research. That's the first goal. And the second is to document the effect of missions of various duration. And the third is to monitor the effectiveness of new countermeasures, such as new exercise or new diet, that are going to be tested between now and the end of the space station. So by collecting the same data, using the same manner over all of these various missions, we have a reference by which we can evaluate the effectiveness of these new countermeasures. So I think I'll stop here and take the questions now.
Great, thank you so much for that. So as a reminder, please dial *1 to get into the queue. It's very fascinating that this involves multiple different investigations and investigators all working from the same data and standards. And it sounds like this is something, since it's ongoing, that applies both to Crew-2 as well as Crew-3. Is that correct?
That is correct. We will also test, we have collected data on Crew-3 during their mission, and we will also collect some measures immediately after they land and for a few weeks after they return to JSC.
Great, thank you. And we have a question from Ken Kramer.
Hey, thank you. Ken Kramer, Space Up Close. I'm curious about this watch that you mentioned. I was at the arrival a couple days ago, and I noticed that Kayla Barron had this white thing on her hand, and eventually I saw that it was a watch. And I'm curious to know if that's the watch you're talking about.
Yeah, I don't know if I can tell the Brian. It's a very, one of these early smartwatch. It's an active watch, and it measures the movement of the wrist of the astronaut. And so this has been used in research, sleep research for a long time. It's called actigraphy. The watch is measuring the movement of the wrist, and by definition, if you're asleep, then you don't move your hand so much. So the researchers can calculate how much during the night, how much is the sleep, and even if the crew wakes up, if they start to move, they can say, oh, there is an awakening here. So that's how they measure, they get a measurement of the sleep and wake cycles.
And one other thing is, if I understood you right, you said you just started collecting this data in 2018. Is that right? I would have thought you would have been collecting these kind of data, boy, for decades already.
Yeah, some of these data have been collected for a long time, but for the first time, we collect them at the same time. So, you know, when a crew is assembled for mission, they are pitched for various experiments, and you don't always have measurements in every physiological systems during an expedition. So as of 2018, we said, well, we should have at least a minimum set of measures that address all of the systems so we can compare the response across these systems. And that's why Standard Measures was born. But of course, we have data on some of these measures from a long time ago. But since 2018, we have simultaneous data across all of the system at once during each of these missions.
And you collect it in a standardized way so you can compare it better, I guess. Yeah.
Yeah, that's the key. That's the key point here, is to collect in the same manner at the same time point and analyze by the same lab with the same method, so we can really see if everything is happening when we change a countermeasures or a diet or something else.
Yeah. Okay. Thank you.
Thank you. And so you mentioned that some of this data is shared also with Dr. Douglas. Dr. Douglas, is there anything else you would like to contribute to that discussion?
No, I would just say that when we're collecting biological samples and we need some of the same measures, it's time savings for the crew, and it can provide benefits to multiple experiments. So we do data share where we can with as many experiments as we can if any of the measures do overlap.
That's great.
Yeah, there is a limitation in the volume of blood or samples that you can collect, so we just collect just what we need. We don't collect twice, for two experiments. We just share the samples.
And then you can use that same sample for multiple data points, it sounds like.
Correct.
Okay, great. Our next question comes from Russell Pound.
Hi, thanks. Russell Pound with Pacific Rim Media. And back to Dr. Douglas. Some of our audience is our school-age students. And in your talk, I think I heard you say, hey, eat your fruits and vegetables and you'll be smarter. I think it came out that way, but just if you could provide some more detail on what are the kinds of foods that you're collecting data on and what kind of results you expect to get back that are specific to what you're collecting in space that helps us back on Earth. Thank you.
Right, and I would say that there is data. So what we've seen is that if you are eating more of these beneficial foods, it can help with cognitive speed. So we are seeing some benefits from things like that. Overall, the kinds of things you want to eat are a variety of fruits and vegetables and different kinds of foods that will provide a lot of bioactive compounds. So it's just really providing a variety and consuming enough. And so filling a diet with those foods also can reduce the intake of foods that are high in sugars because you are filling yourself with the healthier foods. But the goal is to get a lot of different bioactive compounds and to eat a variety of those fruits and vegetables. So it's not a specific fruit or vegetable. It's really just making sure you're getting a high intake, you're meeting the goals. And the goal for this study is at least 6 servings of fruits and vegetables a day. And so that in general, if you eat a variety of foods and you're meeting those goals, the expectation is that you can improve health and performance. And there's ground-based data that supports that as well. So we're trying to collect that with a specific spaceflight food system in flight. And it's interesting, too, because we do have a processed food system, but it shows that you can still eat processed foods that meet those health requirements and still get those benefits. So not all processed foods contain a lot of sugars and things like that. It's the processed food that you're consuming. So, there's a lot of dried fruits, things like that are also processed foods that can provide a lot of health benefits.
And thank you. To elaborate on that, you had mentioned previously butternut squash and kale. Can you share with us some of the other processed fruits and vegetables the crew members have access to?
It really is just a wide variety. So we have even things like Brussels sprouts. We have different types of beans. We have broccoli, cauliflower, and we try and provide them in ways that even as processed foods, they are still healthy and highly acceptable. So where we can, we'll use different spices to make them taste really great without it providing high sodium or things like that.
Okay, great. Thank you for that. At this time, if you have a question for any of our speakers, you can now please join the queue by pressing *1, and we'll take any final questions that we have just before the end of the hour. In the meantime, I will remind folks that you can listen to a replay of this teleconference later by dialing 888-566-0076. And we appreciate all the questions that folks have submitted already today and the interest in this important science, research, and technology. You, of course, can follow along with the research being conducted during the Expedition 66 6-month mission by visiting nasa.gov/station research news, all one word, or following the @ISS_research handle on Twitter. And we'll just check and see if we have questions. We have one more question from Ken Kramer.
Hey, thank you all for doing this. I got a question for Stephanie and Dave. Dave, You mentioned 400 pounds of cargo, 150 pounds of science. I'm curious to know, how does that compare to these other SpaceX Crew missions? Is that standard, or is there more science on this? And Stephanie, I was just wondering if you have a manifest that actually describes, how much of each thing is going up on the station, hardware, science, that you normally do for like the cargo missions. Do you have anything like that for this, and any more detail about the science besides the this Crew-3 sheet from, let's see, October 26th? Yeah, thanks.
Ken, I'm sorry, I was trying to, can you remind me of the reference there for the 400 and 150, because I'm drawing a blank on that.
David, that's in the feature that is posted online. And
yeah, right.
I can't speak to the comparison, Ken, from one mission to another, but there is a requirement in the commercial crew contracts that they be able to provide a minimum amount of pounds for supplies and hardware and research.
Well, could you maybe look into that, or someone look into that and get back to me how this compares to the other flights?
Yes, we can get back to you about that.
Good.
You bet. Yeah.
Good. And about the manifest list, do you have that available?
We'll get back to you about that too, Ken.
All right. I looked, but I didn't see it. Maybe I just missed it. Thank you.
Yeah, we haven't made the same kind of thing that we have for the cargo missions.
Right.
The primary purpose of these missions are the crew members themselves, who also, as David mentioned at the beginning, are also important for testing some of that technology that is that environmental control and life support technology that is important for future missions. So we will see what we can get you and get back to you on that.
Yeah, I think it would be really helpful. Okay, thank you.
And Ken, thanks for bringing this up. I will mention one other thing. We do have a manifest, but it's not even really user-friendly it's designed for cargo and logistics, so it's not like something we can produce immediately and you could read it and understand it. Sometimes we have to go back to the hardware owners to understand the labels that they use on those. So we do have such a thing. It's just not one of those things that if we gave it to you, it'd really be useful. But like Stephanie said, we'll go off and look at this and see what we can do to provide you with what you're asking for.
Yeah, because you do make a really nice 4-page sheet. I think it's 4 pages. Stephanie and I have talked about this before for the cargo missions, and I just think it would be really helpful for everybody if you had something like that for this. Thank you.
Thanks for the feedback.
Yep.
Okay. Thank you, Ken. I don't see any other questions in the queue. So with that, we will wrap up today's Science Telecon for the Crew-3 mission. Please do stay tuned to nasa.gov/commercialcrew for regular updates in advance of the upcoming launch. The next activity is tomorrow evening, October 29th. The SpaceX and NASA teams will conduct a launch readiness review meeting, and that will be followed about an hour after the conclusion of the meeting with a pre-launch news telecon. We're targeting approximately 10 p.m. Eastern Time for that to begin. And it will also be streamed live on nasa.gov/live. And then our launch coverage for the mission does begin at 10 p.m. on Saturday evening for that 2:21 a.m. Eastern launch time on Halloween. So thank you for joining us today, and we hope you have a wonderful afternoon and evening, and join us again tomorrow for the pre-launch news teleconference. Thank you.
Thank you.
That concludes today's conference. Thank you for participating. You may disconnect at this time.